As I've been kindly introduced, my name is Dorottya. I'm a final year PhD student at the University of Oxford and also associated with the UK Health Security Agency. So I'm going to talk to you about some work we've been doing on informing genomic surveillance in England based on results from this national survey we've done called the NEKSUS Study on E. coli and Klebsiella associated with bloodstream infections. So how much is enough? A bit of background on bloodstream infections and why I'm talking about these in an AMR network meeting. So these are essentially used as kind of tip of the iceberg indicators of infection and resistance levels in the UK. Just because they are so severe infections, we think that pretty much all of the infections that do happen end up in hospital and are detected and recorded in our national surveillance systems. So at least for the patient level data and microbiological data, which is what we already do record. The other problem with these is that they have a high mortality and they have been increasing since the pandemic, both the overall numbers of infections and the resistant infections. So the plot on the bottom here, that's just showing kind of climbing resistance, the green showing the E. coli, the orange showing the Klebsiella to the key antibiotics that we would use. So for example, coamoxiclav at the top. And we're talking about E. coli and Klebsiella specifically because these gram-negatives are responsible for a disproportionately high amount of the resistance of all the types of bugs that would cause bloodstream infections. A bit of information about this national survey we've done. It's called the NEKSUS Study. So the National E. coli and Klebsiella Bloodstream Infection and CPE UK Surveillance Study. So what we did here is we recruited 10 sites, so large, busy, acute trust microbiological labs across England, representing the whole country. And over the 6-month period, from the end of 2023, we asked these labs to submit all of their E. coli and Klebsiella bloodstream infections, whether they were resistant or susceptible. And then we sequenced them using long-read sequencing. Now, this long-read sequencing with nanopore is also kind of a novelty of this work because this lets us reconstruct the genomes kind of at a much higher resolution. So it lets us look at plasmids, for example, and also potentially for a lower cost than we would have done with the previous kind of gold standard, which requires sequencing with both long and short read sequences. And this is important because it might mean that genomic surveillance on a national level is a bit more feasible with this method. And I've just shown a screenshot of a preprint we have evaluating or kind of showing this in a small subset of this, showing that doing nanopore only sequencing and genome reconstruction for these bugs is kind of good enough now compared to the old gold standard. So the research question we really want to kind of help the UKHSA answer is how many isolates have to be sequenced to capture a representative portion of the relevant genetic diversity. And now the kind of issue with this is like the two concepts that I've highlighted in bold. So what do we really mean by what's relevant genetic diversity? So is that, do we mean diversity at the genomic level, at the bacterial lineage level in terms of multi-locus sequence type? So this is when we look at 7 key housekeeping genes and we type those and we kind of assign each bacteria profile based on that. Or do we mean at a more granular genetic level, like the types of resistance genes they carry or even the plasmids? The other key concept is what do we mean by representative proportion, kind of what is enough, really? And also, how do we measure this? So do we care about the overall number of unique genetic features there are, or do we just care about the common ones, or somewhere in between? Now, relating to this latter concept, here's just a bit of a reference slide of Ecology 101, so some basic concepts of genetic diversity that are commonly used in ecology. I apologize for all the equations, but the really key thing to take from this slide is that essentially these different measures of genetic diversity vary in how sensitive they are to singletons. So those are species, or in our cases, MLSTs or bacterial lineages that only occur once. So their species richness, which is the most sensitive, where kind of a species that occurs once carries equal weight to one that makes up, say, half the population. Shannon diversity is somewhere in between, and Simpson diversity kind of only cares about the common MLSTs that occur in your population. And I've also included sampling coverage as a concept, which is a bit tricky, but it essentially means what proportion of your entire community belongs to a species that you've already observed. Or kind of another way to look at it is if you have a sample and you take one more, so just one more individual, what's the probability that that individual belongs to a species or an MLST that you've already observed? Just to give a little example of this, so say this is your kind of sample of 10 individuals with 7 species and you observe just one more. So here your species richness goes up by 1, but your other measures, your Shannon and Simpson diversity, increase by much less. And the sampling coverage kind of depends on how many times your pink isolate occurs in the rest of your population. And so why have I just spoken so much about singletons and these rare species? So if we look at the data we've collected, so here these are histograms of, on the left, the MLST types that occur in our data from the NEKSUS sample, and on the right, the different types of AMR genes. We see that most of them are really rare species, so either MLSTs or genes that only occur once or a very few times. And this is kind of more extreme for the MLSTs. So for Klebsiella, 40% of our sample is made up of MLSTs that only occur once, and 32% of the E. coli. And this is a bit less skewed for the AMR genes. So 15% of the AMR genes we detect only occur in a single isolate. So does this matter when we look at our actual data? So here we plotted some rarefaction curves. Again, the orange is Klebsiella and the green is E. coli, and each plot is a different one of our diversity measures. And with the rarefaction, this is basically, we've taken a sample of various sample sizes, so going all the way from 0 to the maximum sample size along our x-axis, and then we've calculated various diversity measures along the y-axis. And the main take-homes from this, and we've repeated this each sample at each sample size 1,000 times to get kind of lighter colored confidence intervals. So in terms of measures of diversity, the kind of important things to take is that species richness, which is this bottom left plot, is quite a poor indicator because it kind of keeps going up, up and up. And you could have almost an infinite number of different species. So if we want kind of a finite sample size of how many do we measure nationally, it's not a very good measure. Whereas Simpson diversity, so that's the top left, is a very stable measure. It's just a top kind of a flat line there. And so we can get a very precise estimate from a small sample size. And kind of sampling without replacement, that's the really light grays. Just as a side note, is probably not a good idea for this kind of analysis. Then in terms of comparing Klebsiella and E. coli, we kind of see that Klebsiella, so the orange lines, are much more diverse, which means that, so from our whole data set, for example, if we look at the bottom right plot now, we've estimated to only have captured about half of the kind of MLST types in the whole Klebsiella population, whereas we've captured 88% in the E. coli sample. And this kind of applies to smaller sample sizes as well. If we just look quickly at the AMR genes or resistance genes, so this is the same setup of the plots from rarefactions, and we kind of observe the same thing in terms of diversity, in terms of species richness being a poor measure, Simpson diversity being a good measure. And when we compare E. coli and Klebsiella, we see that potentially there are smaller differences now in AMR gene diversity compared to MLST diversity. So the green and the orange lines tend to be closer together. And this also means that we kind of get a much higher representation of the overall AMR gene diversity from the whole populations for both E. coli and Klebsiella at much smaller sample sizes. So we can kind of see from the sample coverage, so the bottom right plot, these go almost up to 100%, even for quite a small sample of just 250 samples, which kind of, for context, would be kind of between 2 to 5% of all the bloodstream infections that occur nationally in the 6-month period. And just as a side note, Klebsiella might have a bit less diversity in its really common genes. So E. coli has kind of more very common AMR genes, which is in line with what we know with some E. coli clones having stable associations with certain resistance genes. So kind of that's the most important part. In terms of looking to future work, we know that this is maybe not an intuitive way for public health, in the public health field, to look at diversity. Not many people may have heard of Shannon diversity or Simpson diversity, and we can kind of reframe this question to what we would actually want to look at in a public health setting. So what's the minimum sample size that we need to detect presence versus absence of a lineage that occurs at a certain frequency with a certain level of confidence? And there are luckily quite standard formulae for this that people have used quite a lot during the COVID pandemic. So kind of next steps will be to apply our data to this to get some estimates and compare the various methods. But in conclusion, in our populations of E. coli and Klebsiella, there are many singletons, so many rare lineages and many rare genes. Simpson diversity is a more stable measure of overall diversity, but sampling coverage may also be useful. We know that Klebsiella has a lot more MLST diversity, and the difference is smaller for looking at AMR genes. But a really big caveat to this is limited by the databases that we actually use to label these AMR genes. And the feature work that I've already alluded to, it would be good to look at some other genetic features, including plasmids, look at differences over time and region, and compare this to some other methods of estimating sample size. So that was all I had to say, and thanks to everybody in the various teams that I've been a part of. This is our Modernizing Medical Microbiology group, all in blue for AMR for the WHO Initiative Day. Yeah, thank you, and I'll take questions at the end.